So I can get started while you are promoting everybody to panelists. Well, good morning, good afternoon, good evening, everyone, depending on where you are around the globe. I would like to welcome you all to our second TCBG seminar this semester, which will be presented by Dr. Gerhard Hummer from Max Planck Institute in Germany. Before introducing our speaker, I would like to just remind us of a few important technical aspects of the way we are doing this in Zoom. All the attendees are being promoted to panelists, which means you can turn on your cameras and you can turn on your microphone during a session. So what we are asking is to, of course, turn off the mics in the beginning of the session and only turn it back on if you want to ask a question, which I try to coordinate. It would be nice to leave your cameras on if you're comfortable, to have a little bit more face-to-face interaction with the speaker if you prefer, but that's up to you. So during the Q&A, during the questions and answer, I try to monitor the questions that will be pasted into the chat box, not into the Q&A. You're all panelists, you don't see the Q&A, so you can all chat now. So paste your questions into the chat box. I keep monitoring them with my colleagues, and then we might interrupt the speaker if there is really a burning question in the middle. I hope the speaker doesn't mind. Otherwise, we're gonna actually, at the end of the session, bring up all the questions. And what I'm gonna do, unless we have too many questions, is to ask actually the people who ask the questions to ask the question again in person using the microphone of the speaker, so that will be a little bit more interaction with the speaker. So in the beginning, turn off the mics, leave your cameras on, we recommend, and then at the end of the session, we can deal with the questions and answers. Anything else, Barry, I forgot to mention about the etiquettes? Or
I think you got everything, yeah.
Yeah, we got this. Okay, so let me go with the formal part of the introduction. It's really a great pleasure for me to introduce our today's speaker, Dr. Gerhard Hummer. Gerhard is a native of Austria, and where he did his PhD and initial preparation in physics at the University of Vienna. And during this period, he had also some connection to the Max Planck Institute for Biophysical Chemistry in Göttingen, Germany. So after completing his PhD in Europe, he moves to the Los Alamos National Lab, initially as a postdoctoral fellow, but then later, after 3 years, he became a group leader at Los Alamos during the periods of 1993 and 1999, where he did really interesting work. And then he moves to NIH, to the famous chemical physics laboratory of NIDDK, where he became actually chief of the theoretical biophysics section and deputy chief of the whole lab. And since 2013, Gerhard decided to move back to Europe. He has been actually a director at the director position at the Max Planck Institute for Biophysics in Frankfurt, Germany, one of the famous Max Planck Institutes that especially membrane protein people know very well. And then he heads the Department of Theoretical Biophysics in that institution. Since 2016, he is also a professor of biophysics at Goethe University in Frankfurt. Gerhard has received numerous recognitions and prizes for his seminal work in many different areas. I just list a few of them. He's a fellow of American Physical Society. He was a fellow of American Physical Society in 2005. Received a senior fellowship of the Frankfurt Institute of Advanced Studies in 2015. Recipient of Raymond and Beverly Sackler International Prize in Biophysics, and also Nancy Nostal Scientific Mentorship Award at NIH in 2010. So in terms of research, so we have a lot of overlap with what Gerhard is interested. He is excellent in applying really physical chemistry theory and physical theories to modeling and simulation at a molecular level at different actually scales nowadays. He is interested in membrane protein systems, and recently he has moved to really timely areas of our business, namely modeling of coronavirus, which is something we're gonna hear more from Gerhard today. So it's all yours, Gerhard. So we look forward to your seminar. Thank you very much again for accepting our invitation and making yourself available. We look forward to your seminar. Please.
Well, thank you, Emad. Can you hear me?
Yes.
Sound is good.
Yes.
So thank you, Emad, for a really very nice, very kind introduction. It's absolutely wonderful to be back in Urbana. Even if it's only virtual, I have so many friends there, of course, including you, but many others that I would so love to be there physically, but we will have to put up with with the restrictions on travel. So, as Emad already said, we have so much in common in interests, but also I think the way we approach scientific problems. And I think I've always greatly respected and admired you for your research. And I think, of course, I should say more broadly that it is the Illinois style with Klaus and with Zan and with also the experimental people there that I've been admiring and following for many, many years. So this is a slide maybe in the spirit of showing that MD has come of age, and it's, I think, really become useful. And Emma brought this up. I mean, I've been a student a long time ago. It's now not quite 30 years that I graduated, but it's almost getting there. And when I started out, MD simulation was getting a mature field for simple liquids. That is, we could with confidence simulate argon. And we were kind of starting to get our hopes up that we would understand water. Now, of course, some people maybe also in this audience would say, yeah, water is the least understood liquid, but we have made progress there as well. But really, I think where the most amazing progress has come, in my eyes, is in simulating complex biological systems. And I just start this movie again on the left. This is a simulation that Søren van Bülow, one of my PhD students, did, literally starting out as his warm-up project and grew into something bigger. This is 400 up to 500 all-atom simulated proteins in explicit water, and we could calculate rotational, translational diffusion coefficients, affinity constants, where they interact with each other, and these things actually match with experiment. That is, you have the viscosity of this is a protein solution at about 150 or 200 milligrams per mil, and we get this right. We get the contact things right. In the middle, you see something that we are starting to do now. This is building small organelles and getting them ready for simulation. This is a collaboration with the Sasha Martin's lab in Vienna, where they do amazing reconstitution work on autophagosome assembly. And so we're putting that knowledge into detailed atomic models, and we look at how an autophagosome or an phagophore is initiated, and how this small structure grows into something that really is critical to cellular homeostasis and many other processes. And on the right, this is a paper that is, I think, just appeared online, a collaboration between our group and the group of Achilleas Frangakis here in Frankfurt, who has been looking at cell-cell junctions with electron tomography, and using his information, his data, as well as the wealth of knowledge that has built up, we could start to model desmosomes as mechanically very strong structures, and simulate them again at the level of all atoms, and get a detailed understanding of how transient interactions, relatively weak interactions between these proteins stabilize a rather interesting architecture, strut-like architecture. So this is just to do the warm-up to say, yes, MD is good for something, and I think we can really contribute to experiment. But of course, the challenges have changed in the last few months. I think for many of you, certainly for me, my group, my family, we have all been dealing with a change in our lives. I mean, I hope not in our health state all too much, but of course, even if we haven't been infected yet, many people have, and this has really become a major crisis around the world, both with respect to health, but also with respect to the economic impact. So when SARS-CoV-2 infections became active and we all went into home office, my group decided that they wanted to get actively involved. And out of this, a number of, I think, very exciting projects emerged where we could bring in our expertise in understanding and modeling protein functions, and in particular membrane proteins, but also, as I would show, other proteins. So this will touch upon different aspects of the viral life cycle, as is highlighted here on this slide. And so what you can see here is, this is from one of Brett Glaunzinger's reviews on the viral life cycle. You can see this is a very complex process that is leading from infection to translation, reproduction, to the assembly of new virions, and then to the release of these virions. And in my group, we focused primarily on two aspects and those I would like to share with you in the coming half hour or so. And the first one deals with an interesting, I think also biochemically very interesting aspect of viral infection, and that is how virions and SARS-CoV-2 in particular interfere with our innate immune system. And of course, I think it is now well recognized that essentially all viruses use strategies to modulate our innate immune system. And in the case of SARS-CoV-2, we learned something interesting. And I would argue MD simulations, molecular dynamic simulations, helped us understand how this is done at the molecular level. And the second aspect that I would like to present to you deals with the question of entry of the virus into the cell, the initial step of infection. And of course, we're interested in that because this is a place where with both antibody therapy and of course, vaccines, we have the hope of addressing this major health challenge head on. So let's first look at the immunosuppression that is associated with the function of one of the two proteases encoded by the SARS-CoV-2 genome. And this is the maybe less well studied papain-like protease, PLpro for short. So in the rest of my talk, I will just refer to PLpro, who has multiple functions. One of them is, of course, cleavage of the polyprotein. So it's processing the polyprotein and thereby involved directly in viral replication. This is together with the main protease. And then there is this other aspect that emerged from earlier studies of the old SARS, but now became clearer by looking more carefully at what new SARS is doing in our cells. So this is to attenuate type 1 interferon system and associated with the innate immune response. So this is a collaboration with colleagues here in Frankfurt, in particular with Dong Hyuk Shin, who is at our institute, but also a postdoc or was a postdoc. He has now a faculty position in Korea, but at the time of this work, he was a postdoc here in Frankfurt in the lab of Ivan Dikic. And then the virology work is done by Sandra Cisek and her group primarily. But then there's a long list of authors on this paper with many different aspects covered. So what our collaborators, and this is primarily Dong-Yuk Shin and Ivan Dikic, what they found out was that this protease is very active on ubiquitin-like molecule. And what they found was that it does cleave diubiquitin chains, but it is even more active in cleaving ISG15 proteins. And I have to admit to you, before getting involved in this collaboration, I don't think I paid much attention to ISG15. But this is, as I will show on the next slide, is some protein that is associated with viral infection. So what they found was in a kinetic study is that SARS-CoV-2 PLpro prefers cleavage of ISG15, a ubiquitin-like molecule, over cleaving polyubiquitin chain. And they could also show that the primary reason for this shift in substrate preference to ISG15 is actually just binding thermodynamics. ISG15 binds more strongly to the PLpro. Ubiquitin binds more weakly, and that is the primary factor responsible for the shift in cleavage activity to ISG15. So then you can ask, and certainly I have, what for and why? So turns out that de-ISGylation, this is the removal of ISG, suppresses the innate immune response because the covalent attachment of this diubiquitin-like protein ISG15 to proteins is a part of the cellular response to viral infection. The details, what this actually does, are somewhat murky, at least in my reading of the review articles. Some examples, and I will show one, are better understood, but there are certainly lots of not so well understood things. It's just that we know isolation is one of the mechanisms of our innate immune system to respond to viral infection. So one explanation is that isolation, that is a covalent attachment of this protein ISG, just very in a way similar to the attachment of ubiquitin, ISG would disrupt oligomerization, and it will affect the cellular localization. And in addition, we know that if viruses start to cleave ISG modifications, they will thereby interfere with this immune response. And an example is this interferon regulatory factor 3, IRF3, which is one of the better understood cases, where if it's not modified by ISG15 and in the cytosol, it is subjected to degradation. If it is modified by ISG15, it is not subjected to degradation as much, can be phosphorylated and taken up into the nucleus. From the cytosol goes into the nucleus, and there it acts as a transcription factor that upregulates the machinery associated with the antiviral response. And so if you now have a protease cleaving off ISG15, it shifts this equilibrium between a form that is degradable and a form that is not degradable and taken up into the nucleus, and therefore it interferes with this immune response. So when we got involved, the first structures of a complex of ISG15 with the SARS-CoV-2 PLpro came out, and they show this protein binding with one of the ends sitting in the active site, and the other, the distal one, the end lobe, sitting away from this. And what we could then do is compare the interaction of ISG15 with the PL Pro of the old SARS and the new SARS. And we could also compare the interaction of old SARS and new SARS PL Pro with the other substrate that is well characterized, which is diubiquitin. And what we found was there are essentially only 2 amino acids that change in the interface. One is a change from a leucine to a threonine at the 75, 76 position when you go from old SARS to new SARS, and so become more polar. And the other change is from a serine to a valine at the 67, 66 position, so become more hydrophobic. So that was interesting. And so what we then did is, and this is a work that was done by Laura Schulz and Reza Mehdipour together, they set up MD simulations of various systems. And what they found quite consistently was that the diubiquitin, only one of the ubiquitin molecules stayed bound, and the other ubiquitin, the distal ubiquitin that is distal from the active site, actually starts to dissociate. I think it's in 4 out of 6 simulations of about 3 microseconds length. By contrast, ISG15 remained stably attached in all our simulations. I think these are 6 simulations also of 3 microseconds, maybe 3. So it was clear that these subtle small mutations did have an effect on the stability and also on the relative stability of this. And so we also ran control simulations, and actually those turned out to be very helpful, not just as controls, but also as additional sources of information that gave us insight into what was really going on. The first control was a simulation with a diubiquitin bound to the old SARS. So the way I write this is SCOF PL Pro, so node 2, that means old SARS. And in that simulation, the diubiquitin was stably bound, unlike in SCOF2 in the new SARS simulation. The second control was a double mutant now, where we reversed the change between old SARS and new SARS. We changed the serine back to a valine and the leucine to a threonine. And there, we didn't see full dissociation in about 2 microseconds of simulation, but we saw significant opening up of the interface as indicated here in this plot of a key distance. And it turns out that this was a clue as to what is going on in this interface. What we found was that in the old SARS, you have a hydrophobic interaction between isoleucine on the distal ubiquitin with 2 hydrophobic residues, and one of them is leucine 76 on the protease. In the new SARS, this is where the threonine mutation appears at 76, now 75 position. And when this interface opens up in the new SARS, in all the simulations, the first thing that happens is water molecules wedge in between the isoleucine and the threonine to solvate, in effect, this single OH moiety that becomes exposed. And it is this wedging in, this water penetration, that is often reverting, but in those cases where we see then full dissociation, this is an obligatory intermediate. It is from this intermediate that the interface opens up completely. So this is the first part of the story where the MD simulations provided, I think, useful understanding of why we have this shift to high acylation from ubiquitin cleavage. Another aspect where we contributed was to an understanding of the action of possible inhibitors. So there's one inhibitor that was developed way back in the Mezicar lab when old SARS was still a threat. And sadly, of course, for all of us, this line of research was essentially not continued, I suspect because funding also dried up. But there's an inhibitor, GRL 0617, that came out of this development by the Mezicar lab. So one of the questions is, what does it do? Does it help us? And does it stop the ISG15 processing as well? So we looked at this with MV simulations, and we found that the binding pocket where this ligand is wedging itself in is actually very well conserved. So it stably binds both to old SARS and to new SARS PL-Pro. So that's good. And indeed, experiments also showed that it is a reasonable inhibitor, and actually it has a better performance kinetically as on the protease of the old SARS. So it is an inhibitor of the new SARS PL protease, and that's also very good. But you can see the IC50 is not good enough maybe to go into the clinic with this, but certainly I think it's a good lead compound, and that was confirmed also in virological live cell assays, showing that or establishing that PL-Pro is actually a valid target, because this inhibitory action of the protease activity is almost perfectly in line with the viral replication. So if you at doses comparable to what you need for protease inhibition, you can actually also inhibit viral replication. So that means that this is a target. So this brings me to the second major research project in my group, because then of course that's where much of our attention is, and there's the development of vaccines and antibody therapies. And as I think most of you know, they attack the surface or the envelope protein. So SARS-CoV-2 is an enveloped virus. It has a membrane surrounding it. Of course, that's one of the reasons that my group and I got very interested in it. And it is this membrane contains multiple membrane proteins, and most prominently the so-called spike or S protein that is shown here in this flower-like structure in my cartoon. And this S that binds human receptors, and in particular binds to the ACE2 receptor at the surface of certain human cells that express this receptor, and that leads to internalization either through direct fusion with the plasma membrane or through an endocytic pathway, which is also not so well understood which of the two routes is actually dominating. So when we started out, there were structures of the S protein of SARS-CoV-2 coming out. These were the head of the protein, but we didn't have structures of the stalk and of the transmembrane domain. So those had to be built de novo, and that was, however, not extremely challenging, although there's some little catch that we then later found out that I might briefly mention, because the stalk is essentially qual-qual, where we have very little information on the structure, but this is something that we have been very interested in for other envelope proteins for a long time, is the transmembrane domain. And I will not spend much time on it, although I'm sure that Emad and his group probably would want to know more about this. I think this is still under investigation. But in any case, we assembled a complete model. And then a key aspect of many viral membrane proteins, and certainly SARS-CoV-2 spike, is they are heavily glycosylated. So this is a post-translational modification. And in the ectodomain, this is shown here with these green molecules. So based on mass spec data from the Crispin lab in particular, but other sources, we started to attach glycans, and we also modified the transmembrane domain, which is known to be heavily palmitoylated. So you can see the palmitoyl groups really are very prominent aspect of the transmembrane domain. So this gave us a full-length spike with post-translational modifications. And that, the next big decision was how to simulate this, because it's a rather long molecule, and we were quite in a rush to make a meaningful contribution. So we decided to actually simulate a bigger system rather than a smaller system, because that gives us 4 proteins and essentially linear scaling computational cost. And it avoids also the problem of having to simulate a lot of water. And we packed the protein at a density that was based on tomography data for old SARS, which were available at the time. We ended up with a system of about 4 million atoms. And we were certainly a bit aggressive with the time step, 4 femtoseconds, but compensated that by using heavy water. And in the end, we have now for this system, we have a bit more, but this is what was used for the analysis, about 2.5 microseconds. So if you allow me to do that for proteins, we have 10 microseconds of sampling. So here you see a movie now, animated version of what was happening of a few 100 nanoseconds. And you can see it is actually moving around quite a bit, and certainly the sugars are very flexible. But if you look at the protein, the stalk itself is bending over, and that was very interesting. So if you look at this in this rendering, we could identify between the coil domains forming connecting to the head of the protein and the membrane anchor, that we had clearly discernible hinges that we termed ankle, knee, and hip in our kind of group lingo, but then liked them so much that I think that they survived into the publication. So this confirmed by this hinge-like motion, we confirmed by looking at the flexibility of the rest of the protein when we superimpose the different coils as well as the head or the body of the spike. And you can see these domains are quite rigid, but the rest is really flopping around. And you can see in the angle distribution on the right of my slide, on the MD time scale, we see motions on the range of about 30 degrees. So initially, unbeknownst to us, in our institute, cryo-EM tomography data were collected. But despite Corona, rumors spread to us, and then soon Martin Beck, my new colleague, and I got into touch, and we learned of each other's work. And then we got this first tomogram, and all I can say, it was a fantastic feeling when we got to see that we had actually been on track. So what our colleagues did was they collaborated with the group of Jacomine Kreinze-Locker at the Paul-Ehrlich Institute here in Frankfurt, and this is a card-carrying virology group. So this is what they do. They do virology. They have the S3 labs to do this, and they have been harvesting variants from infected cells. So this is real, not overexpressed protein, but real live variants, and then putting them into the microscope. This was done now in the lab of Martin Beck here at Frankfurt and at EMBL. And then we got tomograms, and I don't know, this is made for the tomographers. They want to see what is really in there. And here you can see slices going through one of their tomograms, and you can see the variants appear and disappear. And I can tell you, these are amazing tomograms, super, super crisp. And you can see features that are just from the raw tomograms very, very clear. And one of the features was they could see our hinges, and they're at exactly the right places. We look at the distances, the locations, and it was just a perfect match. So this was great. And I think this we kind of knew after a week or so of comparing notes and really just taking our MD structures. But then what we could also do was we took the raw tomograms, and Matt Sikora, who was in the lead on the simulation team, could just take MD snapshots without any flexible fitting. So sorry, Emma, no flexible fitting needed. In this one, we just took the snapshots and it stuck them directly into the raw tomogram. And this, I mean, this just shows you the quality of the tomograms, that they actually agree with the ND simulations. No, normally I would say the other way around, but really I think this was pretty good. So that was fantastic. And so confirming the flexibility, and I think, I mean, there's other things that we could talk about, but I want to keep this, my talk on time. So why the flexibility, of course? And I don't have a good answer for that. But of course, I have suspicions, and my colleagues have suspicions. I think it increases the avidity of virus host interactions, which means that if you present your main interaction unit rigidly at the surface, the system is locked in. It has to dock on in just the right orientation at the right place. But if these proteins can move freely on the surface, and this is what these tomography pictures, again, amazing pictures also show, what you can then have is you can have them cluster, and by flexing at these hinges, they can interact with curved cell surfaces, with flat cell surfaces in multiple interaction points. And so what would otherwise be relatively weak interactions, maybe transient interactions, they will start to lock in, and this is what the concept of avidity is, and trap the virion there and lead to very efficient uptake or direct fusion. And the other interesting aspect is that we could look in more detail at the glycan shell. And I don't think this slide mentioned this, that my collaborators, Martin Beck and in particular his group member and postdoc Beata Turonova, they have been able to refine the tomograms. This is, remember, this is live virus tomography in subtomogram averages to sub 5 angstrom resolution, where you can see side chains in some cases. So this is close to a single particle, and they could resolve glycans, again, confirming the extensive glycosylation that we had hypothesized when we built the model. And in particular, they could also see cases of large complex glycans, so not just the little short truncated versions, but real long chains. And that brings me to another point that we're still actively working on. This is the question of, are there good epitopes on the spike surface other than the receptor binding domain that is, of course, targeted by the majority of neutralizing antibodies? And I think this picture gives you an idea, is that, well, there is not much room for antibodies to bind. This shows contour levels of the glycans at relatively high contour level. And here you see at the low contour level, these glycans sweep out large fractions of the surface and make this a very effective shield. But still, we could identify some spots on the surface by looking at accessibility, rigidity, conservation, sequence conservation, bioinformatics prediction, and so on, and identify epitopes that actually turned out to be a superset of known epitopes. So the known epitopes have been identified in our computational set, but we have a few more candidates. The last point I want to highlight before then closing is that glycosylation is not just important on the side of the viral protein, it is also important on the side of the human protein, in this case of the ACE2 or the ACE2 receptor, which is itself quite extensively glycosylated. And what Reza Mehdipour could show in simulations is that these sugar molecules can have different roles. He found that glycosylation at the N90, asparagine 90 site, actually is protective against spike binding. And this is consistent with data on studies in the human population that have found reduced susceptibility associated with this variant, if asparagine is there. Conversely, in his ND simulations with and without glycans, he found that another site, 322, actually leads to strengthened interaction of ACE2 with the spike protein. So the sugar itself, the sugar on ACE2 itself interacts favorably with the spike protein. So you have dual roles possible in glycosylation. So in summary, yes, we have been all sent home and had to work from home office for quite some time. Here in Frankfurt, we have been able to return fully to the lab in the summer, but I'm afraid numbers are picking up here, so we might have to go into home office again soon. But we could still do meaningful, or at least for me, meaningful, maybe for you, hopefully just or only interesting work using molecular dynamics simulations. And so the main points that I wanted to share with you was that with MD simulation, we could gain a mechanistic understanding of how this shift of the papain-like protease from a deubiquination to a de-isolation kinetics comes about, and that water penetration into this otherwise hydrophobic interface plays a role. I also showed you how ND simulations could get us an understanding of inhibitor action and show that this CRL 0617 inhibitor stably binds also to new SARS protease. With respect to infection, this is viral entry. I showed you that the MD simulations revealed a very dynamic spike protein in the envelope of SARS-CoV-2, which is consistent with the electron tomography of our collaborators and colleagues. And we hope that our simulation model can provide guidance for vaccine development as well as the development of targeted antibodies and for therapeutic interventions. It's clear that there is not a lot of room available at the surface of spike if one takes into account the glycan dynamics, but we still found a couple of epitopes that could prove useful. And the last point I showed, and this is probably still fresh in your mind, is that sugars serve an important role in membrane proteins of eukaryotes, and in the case of ACE2, human receptor, they can help and but also interfere with the help the virus dock on, but also interfere with spike binding. So this brings me to the acknowledgement slide, and I want to thank, of course, the people in my group, and in particular Laura Schulz. She did the PL Pro and these simulations with a lot of help and insight from Reza Mehdipour. And the spike and these simulations were done by a team working in an amazing way together. I mean, we had to come up with all the tools and mechanisms to establish collaborative platforms, and the data is just fantastic. This was Matt Sikora, Søren von Bülow, Florian Blanc, and Michael Gecht and Roberto Covino. And the AC2 spike simulation, as I mentioned, were done by Reza Mehdipur. But this work would absolutely not have been possible without amazing collaborators. I mentioned Dong Yuk Shin, who is now starting his own career, independent career in Korea. Fantastic collaboration with Ivan Dikic and then Sandra Tizek, as I mentioned, at the virology. So this is great. And then the tomography. So this was for PL Pro, and the tomography work was done by Beata Turanova and Martin Beck with help from the electron microscopy teams here at the DMBL. So great. And then the virology there was done by Christoph Schürmann and Jacomine Kreinze-Locker and Mühlebach and others at the Paul-Ehrlich Institute. And I also want to thank the various institutions that have been funding this, because again, it would not have been possible. We haven't been able, so there's maybe for you of interest, we got a grant to run our simulations on SuperMOOC, but then because of this hacking event that may have affected computers in the US as well, I believe, actually we were taken off. Oh. And so luckily Max Planck facilities gave us enough computing time to complete this. So I'm extremely grateful for that as well. So thank you now for listening, and I will try to answer your questions. I hope this was interesting to you, and so thanks also for joining and listening in, and I look forward to your questions. Thank you.
Well, thank you very much, Gerhard. This is really encouraging to see that even for such a complex system and process, we can still get some insight from molecular level simulations, and especially when you couple it to experiment. I think that was a big message I got from your presentation, how nicely you can substantiate sort of some experimental observations based on molecular simulations. Thank you so much. We have quite a lot of questions. I don't know, some of them you sort of partially addressed during your talk, but I'm gonna go just over the list. And maybe I start actually having a sort of provocative question. So there are lots of groups that are modeling the spike protein. Some, we have models from Romy's group, from your group, from Bonfil's group. We are putting them together, depending on the density of the protein, how many E's we have, how many M's, lipids, glycosylation. So we're gonna end up having multiple models in the field. Do you have any thoughts on that? Like, I mean, is there gonna be a problem? How should we verify them or compare them or use them all? Or I'm a little concerned personally that we might end up having 10 different models in the field and we don't know which one is right, kind of. What do you think?
So this is an excellent question, Iman, and I can only say, I mean, that when we built the first model, and this we're working on revised models, obviously, but the first model we were flying blind largely. I mean, this was relying on BioArts preprints and very sparse data. Now, the data situation is getting better. I think we have a better understanding, but key parts of the protein, particularly in the stalk area, are still very poorly resolved. So I think this is an issue. But data coming in the community. The other good thing is there is exchange. So we are starting to have a livelier discussion, and I think that will lead to form of consolidation. And the third point I want to make is it is quite likely that there is a lot of variability in the variant and in the spike, and in particular in the glycosylation. If you look at how SARS-CoV-2 remodels the ER and the Golgi, how this processing gets changed. I think that depending on all kinds of variables, I would expect a significant amount of variability, in particular in the post-translational modification. So I think that it is actually, this is the good side of having many models with different descriptions, because they might actually all be capturing a difference element of it. Because it's the same issue with that we had to decide on open versus closed, up or down of the receptor binding domain. And we just said, okay, we want to have one interesting one with, let's say, 1 up, 2 down, but kind of pulled out of thin air. And it turns out this is was a pretty reasonable guess. But depending on the preparation and maybe other aspects, you find more open or more closed. In our tomograms, at the individual level, they were mostly in the down state. But again, variability, but maybe variability that is good.
I see. Thank you. That was really nice perspective, and I completely agree with you, Gerhard. So, okay, so now I'm gonna actually invite Zan. Maybe you wanna ask your question, Zan?
Yeah, hi, Gerhard. Just beautiful work. And of course, I always like anything where you compare it to cryo-electron tomography. I really believe that is the future. One quick question, because you mentioned the differences in glycosylations on the receptor at N90, and I think, what was it, 332. Does that vary from person to person? Because they keep commenting on people with different blood types. Some seem to get it less strong infection. And of course, the blood type is determined by the glycosylation. So do they know anything about that?
Yes. So there is variability as far as I recall at the N90 sites, and there were reports in the literature that I remember reading, and that have. So this was something we didn't know, but then we said, oh, was consistent with our observation. So it has the right change. That susceptibility is lower when N90 is there, and therefore can be glycosylated. But I believe, and I think I looked at that some time ago, this is not associated with blood types. But it is a population type thing. But it, of course, to your question, broader question, this could be one of the reasons why such changes can matter, because even weak interactions with the sugars can help if this were a factor in infection. So if blood type is not just associated with I don't know enough about this with glycosylation of a small subset of cells, but is reflected more broadly in cells that would be infected, that could be an interesting thing to look at.
Mm-hmm.
Okay.
Thank you.
Thank you.
So Nandan, do you still have a question about the role of hinges? I think the match between the experimental cryo-EM and the simulation data was pretty nice.
Yeah. So I have actually like a future type questions. Do we have any like a future plan on investigating more on the what could be the role of the hinges? Like if you can somehow make them like keep straight all the time, some kind of mutations, and do you think you will see any kind of difference in the functional role?
Yeah, so this is a question that I think we are tackling now, both from the computational side, where we look at this in more detail, also what is the extent of motion, because actually the tomography showed a larger range of motion, but it turns out that was because we had restricted our spike to be quite constrained. If you looked at the hinges in isolation, they were considerably more flexible than in this relatively densely packed array. But we're also interested in this, of course, from the experimental side. So So to see now, of course, how variants go into action, I think that's one of the very interesting questions. Of course, there would be nice to have a close-up view of this type of engagement, and indeed if this is happening. I think it would be interesting. This is not our forte, not our expertise, is to look at the amino acid sequences in the hinges also in terms of more bioinformatics type of analysis to see which aspects are conserved in the lineage of coronaviruses and which ones are variable. So again, I think interesting, but that's not something that I would consider certainly myself very strong at. But I would, like, if people in this group are interested in this, I would be very interested in collaborating on such an aspect.
Great. Kieran?
Yeah, I think Gerhard already answered my question. So I was concerned about the density of your packed spikes, which the recent data suggests that they're not so densely packed, but they're more sparsely packed. So that may have affected the dynamics of the hinges, and you may not see the complete motion that you may see with just loosely packed spikes.
Okay.
Yeah.
Okay.
I just want to say something to this, because it's a very important point that turns out that it's more important to consider that the density that we use is seen. So there are patches of virial surface where you see them as dense, but overall they're not as dense. And one of the analyses that we did was to look at the distribution and the clustering of the virions, and I have to say it's perfectly random. The radial distribution function is like a step function. It's like 0 where they clash, and essentially 1 outside. And if you correct for the geometry of the sphere, and the clustering tends to dilute is exactly that what you expect for proteins of this density. So they can cluster, and I think this is important for their, and I hinted at that, for their engagement with human cells. But our simulation system is in that sense atypical, because predominantly they are dispersed.
Thank you. So we have a couple of comments from Dr. Suguna Devi. Do you want to pose your question? Okay. If not available, I'm going to move to Sarthak. Sarthak, do you still have a question?
Yeah. I was just wondering that, like, different strength, are there hinges of different strengths, and does that matter of virus uptake? And second question I have is that, does these spike proteins diffuse over the surface? Do they move over the virus surface?
Yeah, so first question, I at this stage, I cannot say very much. I mean, this is something that we are still analyzing in more detail, because our data was so restricted. So that's why we're looking at other systems now. The second, in the MD time, not really, because diffusion, this is such a big object that the diffusion coefficient is very slow, plus membrane attached. But based on what we see in the tomograms, it's pretty clear that they must be mobile, because their distribution is essentially that of hard disks on a sphere. So if like I mentioned my studies in the 90s, this was a system that I could have studied, hard disks, and I think I did study at some point. And it turns out the best description of the spatial arrangement is, at this course level, is hard disks on a sphere, and that means you would not get that if they were structured by their connections to the inside, so the M protein clustering or other places, but they must be relatively mobile. But I would imagine, but it's possible that that mobility is locked in during assembly, so that we cannot rule out. I don't think anybody, at least I haven't seen data looking at the mobility more directly. I suspect they're mobile, but it's going to be a pretty viscous membrane.
Shashank, do you still have a question?
Yeah, so it's actually related to Nandan's question regarding the flexibility of the spike protein on the surface of the membrane. So I think when you showed this trajectory, the movie, so you have like 4 spike proteins onto the membrane surface. So did you also observe like interactions between the spike proteins because of this flexibility in the hinges, due to the hinges?
Yeah, excellent question. And yes, we did. So that is, they do touch each other, and they interact with each other. And actually, there's just, so I would say, again, this is something that is difficult to sample on this time scale for molecules of this size. So I don't think we can say anything much about the type of interaction, but there was just a preprint appearing, I think, in Science, where they actually report spike interactions in one of the samples. So again, hinting at the possibility that you have some form of stabilization of certain arrangements. But
the last question,
I wouldn't trust our simulations too much. Yeah, sorry.
The last question, Kush. You want to ask your question, Kush?
Yeah, sure. Thanks. Yeah, so I was curious about like if you saw any, if you observed any creation of local deformations in the membrane when the spike protein was creating a hinge-like structure. So because especially because this is one of the differences between the simulation in which you start off with a flat membrane and in the actual case it's a sphere.
Yeah, it's a very good question. This is something that we haven't really looked at much yet. Is the membrane aspect. This is still under investigation. By and large, the membrane, despite this extensive palmitoylation, the membrane did not deform extensively. This is what I recall, but I shouldn't say too much. Because we haven't carefully analyzed that aspect yet. So I hope that with the next simulation model, we will have a closer look at that. Because also then we have maybe more membrane and less dense packing.
Can I have a question also?
Go ahead, Petr.
Yeah, I have a question. How do the spike look like on the SARS-CoV-1? Are they also bending like this? And that's one question. And the other one, what is the density of AC2 on the membrane correlating? How is it correlating with the density of the spike on the virus?
So, hi, Peter, by the way. It's been a while. I haven't seen you in a while. So, yeah. So to the second question, I don't know. Actually, I don't know the answer. So, and I think that I know if it is known. Yeah. And the first question, I think you will have to.
You know, Gerhard, that's actually important because, you know, the multivalent binding which you develop from the spike is basically based on the correlation of the AC2 binders with the spikes, right? And densities must be somehow correlated.
Yeah, so what we do know is that AC2 comes in dimers, so there's always 2 at least. So this is already good. So that, yeah, and they cannot be both bound by the same. Sorry, can you repeat the first question?
Well, what is in spike 1? Do they have also this bending?
Yeah, yes. I don't think this has been characterized because, yeah, nobody did this type of work. But the sequence is extremely highly conserved in the stalk.
So who is going to find this bending spike?
Yeah, so it must be the same.
Yeah.
Yeah. So this is it's higher. I mean, the conservation is really, it's exceptionally high. I was very surprised. I know what the estimate is now for the point of divergence, but it's like 50 years ago. Something like that is what I seem to recall. This is a long time for a virus. So to have the stalk unchanged means something. I mean, obviously, it's the business end, it's what drives the fusion, but it also, I mean, even the elements that are not directly involved in the fusion appear to be quite well conserved. And so, yeah, flexible.
Okay, so let's thank Gerhard one more time. Again, we can't clap, or you can actually clap on the Zoom for this stimulating talk, for for accepting our invitation, agreeing to give us this talk at late evening time in Europe, and really stimulating a lot of conversation and discussion. Thank you very much.
Fantastic. Thank you all very much, and have a good day. Bye.
Bye-bye. Bye.
Thanks, Emma.
Bye-bye.